Helps? Does it help? I hope it helps. We are block C. Who's on their best behavior?
Always on my best behavior.
Well done.
Wait, why? They're both kind of.
Yes?
Just Ali started.
Who said this to you about?
Really?
Nope.
Check up.
Nice. Nice. Excellent.
No, like fully done.
What's that picture?
Nothing.
It's not fully done. You're like fully done, like ready to.
Got this.
Liam.
So,
Perfect.
Why are you there?
Like I don't.
I don't mind, but like, why?
I have to find out. I also have to put my stuff back. But it takes so long. You have to like work on it.
the reels I watch, oh god.
Okay, you should be open to this page where we left off last day, I believe.
We weren't here last day. Okay, I was able to find it. Protein synthesis.
Yeah, we're there. Take this moment, double check your work. A goes to
T.
you. For RNA, T goes to A, G goes to C, T goes to A, G, U, C, U. So, a reminder, RNA always starts, what replaces the T in RNA is U. However, T bonds to A. And that is a source of some... Yeah. So check yours. It's also being recorded.
You just change for mRNA, you change the
Yeah.
T in the bottom section.
Wait, can you go back to the...
No.
Wow.
You've already pre-done it. You're just checking, right?
What?
Go. Check. While you're checking those, go through, check the DNA. So A in DNA, A bonds with
T.
T. Yes. Sorry? You forgot your... Yeah, you should be running. Run like the wind. Okay.
What were the last three dissections?
Julian, are you informing me that you're not paying attention to class? Excellent. Once you have the DNA, that is your reading strand, and then you made the corresponding RNA. T goes to A goes to U, C goes to G, and so on and so forth. What should this strand look almost identical to? Not the reading strand. Yeah, the one above it. Because they do match. As long as you replace the T's with U's, you can just go along A, U, G, G, U, A, G. So as a quick double check, C, apparently don't know how to make a U. And that's your mRNA. What is this process called? It's got a magic name. So it's a T, yes? This is transcription. So that is the first stage of protein synthesis. First, we have to get the message, the code for which protein we're going to make. We make it from a portion of the DNA into the mRNA. Then, anybody still reading there?
Reading.
Oh, just to allay any concerns with the less people copying done. It does appear that the Privet plague is not actually becoming a plague, and it is decreasing. There were fewer people sick this morning, and more colds and less vomiting. So I live in perpetual hope that the plague will strike a different house
Ooh, vomit
and
on the
strike it
floor.
down as bad as we got it.
Oh my
Because
God.
I feel like someone else should experience that.
Mr. Neufeld,
Yeah.
was it last year this time? Or was it
About this time, because what happens traditionally, everybody comes home from break. I will not name them, but one particular boy was dropped off by his parents while vomiting.
This
And
year?
so that may be patient 0.
Yeah.
And last year, at one particular day, we had 62% of Privet boys on either dorm rest or in the house. And over half the house was not in class. The hallways, you could almost see the miasma drifting down because you could definitely smell it. The smell of vomit was like thick everywhere, and all you heard constantly was someone in the bathroom just throwing up. One person came to see me, and they were like, "Mr. Neufeld, I escaped." And I'm like, "Well, what do you mean you escaped?" "Well, I was feeling sick, but then I threw up, and I feel so much better. I think I escaped it." And I just laughed and handed them a bin, and they were up all night, got no sleep, just vomiting constantly. Because that's how it starts. You feel sick, you throw up, you felt better, and then you vomit all night long.
Do you remember who that was?
Yeah.
It might have,
Can we get names?
I remember I said something like that.
That experience happened to a lot of people. But in this particular one was special to me because it was Wilbur.
Mr. Neufeld.
Yes.
I think it's still like prominent. It's
Yeah,
just
it's pretty prominent.
That's just
Oh, there's a lot of people feeling ill.
They're just going to
Um,
school.
they're just
stuff,
going to
they need
class. Like I don't
And
feel
if-
good right now.
as
I'm
long
here.
as you are washing your hands, and number one has been shown by research over and over and over again, as long as you're not doing something stupid like licking other people or eating the food after they've eaten it, don't touch your face. Your eye has very few cells from your immune system in it as a primary source of infection. If you touch something and then you rub your face and then that gets in your eye, that is a good way. It's nice, warm, mucousy membrane, and viruses like to slide right in.
I just rubbed that out.
And one would hope that the people who are sick are wearing a mask and trying to prevent other people from being sick. But as we know, there is a certain social penalty to doing that, and so some people opt not to, and that's fine. Just wash your hands, protect yourself, you'll be good. I did not get sick last year, despite arraying around all the boys, until I had to literally scrub vomit off the carpet, and that took me out. And I did it without a mask because I'd run out of masks.
It ain't easy, dude.
So a brush on carpet flicking up stuff into my face.
It ain't easy, dude.
Yeah. So, for the boys. Okay. We're going to start with an overview of the second phase. We had transcription. What's the second part called? Translation. And reminder, anybody who's feeling sick, who's in Privet, please cough on people in other houses, not our house. So, mRNA
Are we sick or sick?
leaves. Where are the ribosomes located?
Rough
Rough
year.
ER. So go to a ribosome. In rough ER, they also float around the cytoplasm. So either would be good. New type of RNA. We're going to talk about tRNA.
Wait, in the RNA cyto.
Plasm. Cytoplasm. Cytoplasm. Plasm.
Cytoplasm.
Cytoplasm. It's the cytoplasm. I just like the sound of that word. That's a terrible word. Maybe if I group it first. Oh, beautiful. Yeah, I'm pretty sure it's a spell check. Cytoplasm. tRNA is transfer RNA. So you have here the messenger RNA, or the mRNA. And this is going to be I couldn't possibly have messed that more if I tried. mRNA. With the code along it. The code is read, if you noticed, in 3-letter sections. Those 3-letter sections are called, oh, no, that one. mRNA has 3-letter segments. They're called a codon. So the code is made of codons. Each codon codes for one piece of the chain. One codon codes for one amino acid. Oh, I should have asked that first. What's an amino acid? Protein. It's the building block of protein. Some people get like large tubs of powder and then they mix it in with stuff and then they drink it before a workout. It's just amino acids. When you eat food and you get the amino acids from the food, you're either eating like chicken or chickpeas, something high in protein, and you will take their protein and you'll break it down into the amino acids. The amino acids are the fundamental building blocks of protein. We should probably put that in somewhere. Amino acid. Protein building block. Julian.
So, that's picture is mRNA.
So, mRNA is the green section. tRNA are the yellow blocks. Also, it's spelled wrong. So I will now correct it. That's not probably a good color. Define custom colors. Need to lighten it up a little. Sure, about there. Okay, so I'm going to correctly spell tRNA because clearly this was. So I picked this diagram because it's a little colorful. It's actually a really good diagram. I shouldn't. But tRNA is spelled tRNA, not tRNA.
Yeah.
You could correct your diagram. What's the difference in spelling?
Lowercase.
Lowercase t, we always do a lowercase t to emphasize that's part of the function, not part of the molecule. The molecule is RNA. T is transfer. Yes?
What's the red bit of pink bits?
Ah, pink bits. So those, good question, are amino acids. So each tRNA is attached to one amino acid. It floats in, and it has a matching code. When you have a matching code that allows it to bond, what kind of code is that? What kind of pairing? Complementary. That's what we say when we're nice to people. It's called complementary base pairing to the mRNA. So this right here, this is a codon, but this is an anticodon. Yeah, it's an anticodon. The matching based to mRNA, tRNA matching based called anticodon. There's something wrong with that sentence. tRNA has complementary base pairing to mRNA. tRNA matching
Base.
bases. I think that's what that was supposed to be. I find trying to figure out sometimes. Ooh, no. What? There. That color. Excellent. Now no one will know.
Unless it's like
No one will know.
you still need to recognize it.
No.
What's the point of the ribosome? Like, why does the mRNA and the tRNA need it? Like, why can't they just do it anywhere else in the cell?
Oh, this is a good question. And it comes back, why do we have a classroom? Because really, if I'm here and you're here, we could do this anywhere. And it just provides a space for it to happen. And so there's a few things the ribosome has. I haven't mentioned. There is it down here? There's the last type of RNA. The last type of RNA and its function. The last type of RNA is rRNA, or ribosomal. And it helps the ribosome stick to mRNA. And so the ribosome sticks to the mRNA, which provides a space, and then the tRNA has a space to come in and stick to the mRNA. It also helps with the signaling of when to stop. So because we're here, we know when to stop. Can you imagine if we did class in the middle of a forest? There would be no bell, and I would just teach forever. You'd be there and be like, it's been 3 days, Mr. Neufeld, we haven't had any sleep. And I'm like, the bell hasn't rung yet. And we would just keep going. When I show you how it stops, there's actually an interaction between the rRNA and the ribosome that actually breaks the whole process up. Yep.
What does that say? Anticodon?
Anticodon. So we have the codon and the anticodon. It's like you have pasta and antipasta. And if you put pasta on antipasta together, your dish blows up. That joke may only make sense to me because for years I thought antipasta was actually antipasta, like the opposite of pasta.
What is the...
That is not written well. There we go. Julian?
It says ribosomal RNA helps ribosome something.
Stick.
Okay.
Oops. No. It lies. There we go. Function of the tRNA. It just carries the amino acids to the ribosome. So brings amino acids to ribosome. Is there anything I've left out? So which is the actual method? The tRNA with the anticodons or the mRNA with the codons?
The tRNA.
Why?
It's a copy of the reading strand mRNA.
Oh. So it's a copy of a copy. I should show you is it multiplicity? They invent cloning and they copy Steve. And eventually Steve is a copy of a copy of a copy, and he's not so good. He puts pizza in his wallet for money. What's the job of the tRNA? Transport amino acids. What is the job of the mRNA?
Have the message?
Have a message. So the coded message is the mRNA. Yep.
How does the tRNA know where to go, so to speak? 'Cause anyway,
Oh, yeah. I like that question. Good question. Any other questions?
And this is all inside a ribosome?
This is all happening, yeah. inside or around the ribosome. The long answer is there's probably some sort of movement function with the, remember the long strands we saw with the motor protein moving around? We think a process like that might be going on. We actually really don't know a lot of detail, but that part is actually starting to come together now. When I was in school, they kind of knew that how it was brought there. It sticks to the ribosome if it's the right one in the right kind of way, but how does it know to let, and like, do they just randomly hit it? At one point, they just thought they all hit it, and the ones that don't stick just float away. That would work, but now they think there's a bit more direction that might actually be bringing them to it. Oh, okay. Okay. Which is the coded message, tRNA or mRNA? It is the mRNA. And you would be forgiven, because the reason I asked for that is for the exact same reason that it was said tRNA. Yes, they're both kind of the message, but this is the overall message. That is the bringer of the stuff. So the chef plans the menu, but the wait staff bring the stuff to the table. This is like the wait staff. This is the chef. So mRNA should be, as far as you know, because I'm lying to you, a faithful copy of DNA. A portion of the DNA, but of the DNA. Now I'm just going to show you something. And why is your laptop up? Put it away.
It's been open the whole time.
Is what?
It's been open the whole time.
Yeah, I know. Telling me I don't pay attention to things is not just put your laptop away. Put it in your bag. Good.
Wow. Okay, good. Anybody still writing? Turn to this page. You have a blank page. Looks like that. I'm going to take this down. We don't need that one anymore. Stop.
Is it just
In
this?
the name of love. Yep.
Why is it blank?
You're going, I just want to show you something. Don't write anything down. Okay? So, we have here, is this top one DNA or RNA? DNA. How do you know? Yeah, it's British. It has T.
I like that one, dude.
Okay, so then we make a copy. That was a process of transcription. And then the mRNA floats out into this cytoplasm. Watch it floating. And then a ribosome comes in. Boom! A ribosome is made of two parts. A ribosome has a small subunit. You don't write this down. And a large subunit. Don't write this down. And then a tRNA comes in. If this is the codon, what is the anticodon? It, UAC. So I'm going to go down and grab my UAC. Here we go. Yeah, it's in New York. If you've gone to the Met.
Why is it called that though?
I'll show you one in a moment. So, the tRNA, so I'm also going to say that there. tRNA, that's a T, lowercase. And then that is an amino acid, and in this case, it is methionine. And I'm going to show you how I knew that. And you probably thought I'd never say this again. Get out your data booklet
I don't have that.
and turn to the biology page. If you need a data booklet, I have more. Come on up. They look so pretty.
Look at the throw here.
The thing you do for chemistry.
It's here. This is all you need.
Okay.
Okay, where's the biology page?
Very bad.
Very back, it's last page. And look what it has. Oh! It has all of the code. Now, does this table use codons or anti-codons? Read the table. Read it. Oh, you don't have.
Hold on. I thought I had it, but I think I lost it.
That's okay. I have lots.
Wait, no, these are
It
SS.
says
No. I want you to
anti-codon.
read the title of the table.
Codons.
Messenger
Codons. They
RNA
are
codons and
So the codons. You will be tempted when I say, what is this amino acid? And half the people will look up UAC and they'll get the wrong one because it's not that one. Which one is it? Which one do you look up? AUG. So you look up here and how this works is this is the first letter, A. And then you see where it says second base, AU. And then in the box you see all four types, AUG. AUG codes for methionine, met. There's a star beside it. What does that star mean?
AG is an initiator codon.
It's an initiator codon. AUG is always start. Every single peptide and protein in your body starts with methionine. It is what signals start here, and it codes for an amino acid. This phase, and I'm going to get rid of the DNA because that's over in the cytoplasm, don't need it anymore, is called, in protein synthesis, initiation.
Can we write this down or no?
No, don't write this down.
Why not?
The ribosome attaches to the mRNA. The initial tRNA start codon attaches to the mRNA in the ribosome. It sets up. This has a. If we're in biochem, then have more names and there's other things. But this is good for us. It's actually good for biology 12. And that's called initiation. You need to make room on your paper for 3 drawings of approximately this side. We'll write up to here. We'll do some labeling. They'll get more complicated. Now draw it.
What's that?
Yeah. Draw this and write all the words down.
Should I draw it?
Yeah, now.
Okay.
Yeah. Make sure you have room for at least 3 of these drawings. Because if I said draw, back when I was talking, you wouldn't listen to me talking. You'd be drawing. And teachers become teachers because they're egotistical actors who just want to be in charge of a captive audience who can't lead because they're failed actors.
Yeah, that actually makes sense.
So do I have to draw like the whole AUG?
Yeah,
G,
yeah,
Like all the way
yeah.
to the.
Yeah, all the way to the answer. Yeah.
Do we need
And
more,
if you can color code, that would be good too.
do we need more ribosomes?
Not yet. Oh, no, there's only one ribosome. But you're just drawing this. Leave room for 3 complete different drawings. You're not going to draw on top of this. You're going to draw it again.
What?
Because you can animate what you draw. So we're going to show it in stop motion.
Can we make like one of the flip booklet things?
Oh, that would be so cool. It would be cruel if I made that as an assignment. You must make
Just
a
get
flip
like,
book of all.
just get like these students to do like
Yeah.
pages.
Oh, yeah. And once again, I wonder if I said color code. I did have pencil crayons, and I'm pretty sure someone came in and stole them.
Shoes.
We need to. Too much hands can be just like all of that.
Oh.
I will say, if someone has an idea like that, like make a flip booklet, that's easily worth extra marks. You just make it.
I know what we're gonna.
A reminder, you should also be studying for your
Quiz.
quiz. Your quiz is not on this stuff. Your quiz is on worksheet 1 and 2, which is just before this stuff. I posted the answers, beginning of prep and middle of prep for 1 and 2. In addition, your quiz will be open workbook. If you want to put on your worksheet explanations or more detail on your worksheet, you're welcome to. It is open worksheet, so if you want to know how to spell certain words or stuff, because the questions will not be the same. I don't want it to be like, oh, I copied it. Was your first block teacher talking about AI at all today?
Yeah.
Yeah.
So, we all had Pro D this morning about AI. And so
Yeah, it's cool.
this is my particular method. I'll ask you different questions because
Is this
Um, I don't want to mark AI.
is this good? Or is it too messy?
No, it's up. Can you read it?
Yeah.
Yeah. The only thing is that that is not bonding.
Oh. I just did it so in space.
Because we, as AI, we are living in perpetual fear, not no fear.
Yeah.
More like, we have to make sure that you learn. So if you're using AI to do the task of trying to train your brain, you've cheated. If you use AI to leverage your brain to accomplish a task, that could be brilliant.
You literally just write that
I
down.
could actually show you one
Oh,
of
you
those.
see ribosome there, but it's cool.
But yeah,
This is
see,
a ribosome, so
I,
why is it
I use AI
It's a
now.
You're welcome.
Why?
What's a peptide?
Good question. We did it last night. What's a peptide?
I was better.
Hold it. Where is it on here?
I don't know.
It's like that.
I don't even know what a peptide is.
Oh. No, but you've asked it for a reason. Where on your notes do you do that so I can write it in the same spot?
I was thinking the peptide was like near the amino acids. Yeah.
Yeah.
The peptide chain.
Brings them in.
The growing peptide chain.
Growing peptide. Where is that saying? Growing peptide chain. A peptide is a string
Oh, got it.
of amino acids. One string of it. So essentially, it is a protein. It is a small protein. The reason we make the distinction is some proteins are made of multiple strands and are larger. So a peptide is what we're coding for, one single protein strand. Does that make sense?
Understood.
Yeah.
Mr. Newfield,
Anybody still drawing?
wait, I
Yes.
have a question.
Yes?
Yes.
So, you
Speak
know,
up there,
when
buddy.
the tRNA chains in with the mRNA, how does that specific chain know exactly how it pairs and what protein to bring? There's no way the odds.
Oh, no. UAC is an anticodon.
Yeah.
That tRNA always only binds to methionine.
Okay, but then, like, is there always by chance going to be like the next one that's gonna always pair with the next
They are all floating in the cytoplasm.
Oh, individually.
Yeah, individually. So it'll just grab the right one. Right? If the wrong one comes in here, say by chance, it's not going to undergo complementary base pairing. It'll get kicked out. Yep.
Where are those made? Like the anticodons?
The anticodons?
Like,
They are themselves are made by enzymes. Yeah.
Also, do they have to like go into the mRNA in order, or can they be like?
No. They
MET here. and
one
then
no.
here?
They have to go into the ribosome. So it's not going to happen over there. All of this happens in the ribosome. So the ribosome provides the place for it to happen. Just like this classroom provides the place for Science 10, you would never learn any science outside of this classroom, right? Only here.
TikTok.
None of you are like in like watch, read, hear podcasts in another like marine biology class,
Watch TikTok.
or in any one of those. You only do your science here because it only happens here.
I watch TikTok.
Because it would.
I watched the Joe
Pardon?
Rogan
What
podcast.
about
The
the
guy is
people
a
that want to drop
scientist.
dead balloons outside?
Neil deGrasse Tyson.
Oh, I just took the classroom with me.
No.
No, he didn't.
Yeah.
So
Yes.
what if the ribosome
That
has
aspect
multiple
of physics
codons in it, like more than two? Then
The ribosome is drawn this way. It only has two sections.
I know,
Oh,
but
so
I'm
we
gonna
only
have
hold
to.
two.
Only hold two. Okay. We good? Okay. Stop writing.
That's
Look this way.
I'm done writing.
Okay. We're gonna do the next phase because initiation is over. Go away. So now the ribosome. Why did it did it select the whole thing? Yeah. What? Oh my God. Yeah, nothing is selecting. What's going on?
Do you select the wrong thing?
I'm gonna save it before it crashes. No, it doesn't. It's trying to text recognize things. Stop it. it's selected everything? Edit, undo, move. Okay. It's still doing it. What the heck's
What
going
are
on?
they? Hearts. What heart?
Recognize, change, remove, link, sound. There.
Sure, it's a heart.
What the? I'm gonna get, you've already got them labeled, we don't need to label them every time. Goes there, there, okay, now I can move things again. We're not moving it. Next phase. What is the anticodon we need? If the messenger's GGC, we need, yes?
CCG.
Excellent. And using your table,
Protein.
what amino acid?
Glycine.
Ooh, we got two different ones. Which CCG is? Glycine.
Oh, it's is it not?
Everything is GPS.
Isn't it CCG?
CCG is the, what kind of codon?
I just see proline.
No, look at me. Julian, look at me. CCG is the anticodon. What type of table is that?
Oh, is this for the codon?
That's a codon table, so you have to look up GGC.
Where did you get this from? Was this
where-
handed out while I was at the bathroom?
We all have it.
This.
We all had it.
Look. Data table,
Glycine.
back page.
I'll get, I'll get it.
There you
Thank
go.
you. It's a good feeling.
It's yours. Good?
Yes.
Okay, don't go to sleep. No sleeping. That would make me feel bad. Okay. This comes in. A bond forms between the amino acids. That is a peptide bond, which from your purposes is just, it's a special type of covalent bond. But as you'll learn in biology 12 and maybe 11 if they do this, it's considered special. And it's just because it has a hydroxyl on one side and anything there, but it's not a big deal. But they call it, it's a magic word, peptide bond. And then it moves over, chunk. Once it moves over, this tRNA, which we don't need that label anymore, you've already written that down, that tRNA floats away, it's done its job. It's served the dish. We've got the amino acid bonded to the other amino acid. We've got room here. What's going to attach to GAU? CUA. And what will that code for?
Aspartate.
Aspartame. And that floats up and goes chuk-choom. I've got to provide sound effects. I was trying to apply sounds and it didn't work. And then what would form in between there? Peptide bond. And then it would, okay, I've probably done enough that I can now, this part is not initiation. This part is elongation. Now please draw that.
What part is this? Is it elongation?
It's elongation.
With CRISPR?
No CRISPR. I mean, we're reading a gene that you could have changed. CRISPR happened before this.
Elongation?
Yeah. If you're doing stuff here, you're doing it post mRNA
Fruit of my knowing.
editing.
My
It has a
spawn.
special name.
Where
Actually.
does CRISPR even happen? Like, can we do it here, or is it like, what do we need?
You need a lab and a patient. Not that kind of lab. You need a biochem lab.
Okay.
Doctors.
I'll be the patient. Can I sign myself up as like a test subject?
Yes, I believe supervillains worldwide are looking for test subjects. I do have my advanced
Mr. Neufeld cloning virus.
I have a
I'm
question.
learning.
I have a question.
Would you like to be the first clone to Mr. Neufeld as your body slowly morphs into mine?
No, I really would not want.
I think you just volunteered. Yes.
No,
No.
I just need the fireman thing today. What do we do for the fireman thing today? What do we do?
Oh,
Where do we go?
yeah, I'm about to text you all.
Mad, how?
No. He has refused all of the volunteers. Not because he doesn't want them.
Because we're sick.
The amount of response from the whole school, he has way too many volunteers than he needs. Because when people heard you can miss prep and act like a jerk and be the hysterical friend, they were like, sign me up. And Privet House is sick.
No, I'm not sick anymore.
No, he doesn't care. He doesn't want anybody from Privet.
Oh, is
Oh,
this?
who is this? Who is this guy? Who is this
Who
bum?
guy?
Mr. Chris, he runs
Chris?
maintenance.
CRISPR?
Not CRISPR, Chris.
Oh, Mr. Crisp.
We
We're gonna have to
need
email.
more
What's
hysterical
can we
people.
email Mr. Crisp? What's his last
Pardon?
name?
We need more hysterical people.
No, he specifically emailed that he doesn't want anybody from Privet.
We will be emailing him about this.
No, he just has enough people. He actually thought like if he asked the
No,
whole
we need
school.
more now.
Really?
Yeah.
Okay, I'll let him know. If he
What's
gets back to me and
the
says
score?
something,
Can
I
we
will.
first on the
I'll
list?
do it.
On the
He
list.
has to be
Yeah.
hysterical. For
For
what?
senior play.
You have to pretend to be
Just
injured.
this evening during
I'm
the
very
class,
good at
like,
acting.
if,
I'm technically
fire
injured 'cause I am
department did,
sick.
like,
You don't say that.
drill, I have
You gotta think.
I have a broken spinal cord. I cannot move.
Yeah, you're being like, it's a disaster scenario. So
I'm a
you
good
need
actor.
people pretending to be injured and
Am I gonna
hysterical
get, like, picked
friends.
up?
No way.
Like, am
We
I gonna
once
get
did-
like picked up by a fireman?
Do I want this?
Is everybody done writing this down?
Wait, no,
Fire
it's
people
not-
aside.
Wait, I have a question. Where is this gonna be held?
Okay, again, you're not going. I'll tell you if you get going.
Can you make an
No.
appeal for us?
No,
Can we
Is
make an
it
appeal?
mandatory?
no.
We will be. How will we email
Again,
Chris?
it's a volunteer, so you don't have any rights to go. There's no appeal. He just doesn't need
Well,
you.
now I have the rights to volunteer.
And it's also not like he doesn't like you. It's just like
Like, we
right
didn't even
now
get to
you're in the
audition.
house that was a little sick.
We can
We
audition.
get to audition. Like, can
Where's
we
our
go?
chance?
You literally told me you were sick.
I am sick.
You can't go.
Can wear a mask.
He doesn't want you.
I have been.
Like you are the person he doesn't want.
I'm not sick, dude.
I don't
Sign me up.
appreciate that, Mr. Neufeld.
Yeah.
Is that racist?
What?
Never mind.
It is a form of
Can
discrimination
we be the first people
against
on the
sick
list,
people.
though? Like,
Okay,
can
no,
we
stop.
message?
We're spending way too
I
much time
feel
on this.
betrayed.
Anders, focus. Are you done this?
Yeah.
Okay, are you done this?
Yeah.
Is everybody not done this?
What's the?
Keep
it's-
going?
I'm
Okay.
done. I'm done.
This is shocking, dude.
Mr. Neufeld, I would not goof around unless I was
What?
done.
What
You're-
does it say? Peptide what?
I would prefer it if you said,
Peptide bombs.
if I was able to tell Mr. Crisp that he would never goof around, that would be better.
No, I'm talking about like I wouldn't have talked about the firefighter thing
If we weren't done with our
if
work.
I wasn't done with the work.
Oh, I see.
But this.
But is everybody done with the work?
Yes.
If
No,
we're allowed.
and we're disturbing
if
them.
we're allowed.
Shh, shh. Yes, you have to write all that down.
Oh my god. Christmas is like, oh.
Everybody wrote all this down, right?
Oh, what?
Yeah.
Christmas is good.
That's what elongation
I
is.
didn't, I didn't get the first one.
Oh, I can go back to it, but we get this one done.
Okay.
If you run out of space, you could get some more paper. It's like when someone says, if I'm not back in 10 minutes, wait some more. If your teacher doesn't show up to class after 15 minutes,
I'm leaving.
You can go?
you have to stay there.
I'm still leaving.
And technically, the class sends a classmate to Ms. Allen's office and let her know you don't have a teacher, and she will provide you with one.
Yes, obviously.
If she can't, she will come down and teach you herself. At Brentwood College, there is no rule that says you can leave if the teacher's not there.
Have any students left though?
All the time.
Yeah, why would
Because
a
everybody
teacher?
thinks it's 15 minutes.
Why would a teacher be 15 minutes late to class though?
I have been 45 minutes late to class.
You should really send an email about that.
Because I'm in Mr. Sullivan's office with 3 other boys.
Ooh,
Who was that?
What did they do?
I'm not going to tell you.
Why not?
And that is why we don't allow students to go up under the bridge anymore. No, I just made that up entirely. We don't want you to go to the bridge because there's needles down there. We don't want you to poke yourself. You just imagine a grade 9 going, what's this?
What if there's like a drop?
I can see
Like
the red
both
zone
of
do
them.
that.
What?
You mean the bridge over there in the forest?
The bridge that the cars go over and you can go underneath to get up to the Sally Ann.
I didn't even know there was a bridge till there's an email about it.
Yeah. That's part of the problem when we tell people things is that you then inform them on what they could have done.
Why
And then
not?
we're like, should we do that? It's like when we had, when we tell you not to steal alcohol from the lab, it always becomes a speech because then students go, there's alcohol in the lab? And then we have to tell you why not to steal it because it's literally poison. It's not the alcohol at home in your parents' alcohol cupboard that you could steal that would be much safer. Because it's not drinking alcohol. It's not ethanol, it's methanol. And ethanol turns to vinegar in your liver, and that's safe, and you pee it out. So ethanol requires quite a high amount to be toxic on a permanent basis. You can drink to the point that you pass out and stop breathing, but it's a little bit more. Methanol doesn't turn into vinegar, it turns into formic acid, which is ant bite and a nerve toxin, and first you go blind, then you'll stop breathing, and then you'll go into convulsions and you'll die. So don't drink alcohol in the lab, it's not safe alcohol.
Someone
Worth
done
a
that.
shot.
Someone stole methanol from the lab, like methanol, like the stuff that's not
Did you
safe.
drink it?
So we went in front of the whole school in assembly and said, this is why. If you have it, please turn it back in. And if you're too scared to turn it back in, we just want you to be safe. Pour it out, don't use it because it is literally
That's
unsafe.
very
And I went
stupid.
through why. It's for the same reason airlines still have ashtrays in the bathroom. You're not allowed to smoke on a plane. It's never ever smoke on a plane. But they put an ashtray in the bathroom for the person who does. If you take your lit cigarette and you put it out in the bathroom and start a fire because there is no ashtray, that's even more unsafe. So we have to have a mechanism for people who are doing unsafe things to do it more safe.
I mean, is that like safe injection sites?
That's exactly the theory.
I still
That
think
is
that's
why
stupid.
every house is supposed to have a condom prefect. You're not supposed to be running around having sex everywhere. But on the other hand, if you do, we'd prefer you didn't welcome a new child in 9 months or have an STI. No, every house is supposed to have one. Yeah. Yeah. Actually, I have to check in with our condom prefect. He hasn't gotten new ones in a long time.
Leo
Either
actually gave me some.
he's forgotten he's the condom prefect or nobody knows who he is.
No one knows.
Yeah, who is it in first?
Leo.
There we go.
Leo Cole.
I'm not even sure he got the first round. Okay, everybody done? Yes, question?
He has.
Oh, your hand is up. Anders, attention. Good. Okay, so I'm just going to do, this is just the elongation again. I'm going to take away the notes. We don't need that for now. So elongation happened.
Ooh.
There we go. So moves over again. Chunk, chunk, chunk. This moves away. Peptide bond that was formed. It moved over. What one will this attach to?
That's in
Which, what would the anticodon?
CCC.
CCC. Do you put a P on the end of that? I think it's Russia.
CCP.
There it is. And it moves up. I'm going to grab that. Comes up. We form, what kind of bond do we form?
Covalent bond, peptide bond.
Peptide bond. Elongation. Now. Chunk, chunk. Please look up. What does UAG code for?
Stall.
Stop.
Terminate
Stall.
code.
Yep.
Stop. No.
I like it if it's the power of friendship and we're stopping in the name of love as well. That does not code for an amino acid. It's a stop codon. It interacts with the rRNA that's in the ribosome. So there's a little interaction there that actually disrupts the bonding, and the whole thing just flies apart. So this disrupts that moves away. So the two subunits of the ribosome disappear. The mRNA moves away. The tRNAs come off. And by the way, this can now immediately restart. Like you can get a new one here, and if this is long enough, and many of them are, you'll actually get a ribosome moving down, and another ribosome will come in behind it, and you have multiple ones going down. It's called a polysome. We don't have to worry about that. But it could restart down there. But now this chain, you have made a polypeptide chain. You've made a peptide. And that, now, if it's an enzyme, it's going to go start doing its job. It's going to catalyze a reaction. If it's the little motor protein we saw carrying a vesicle in that animation, it's going to go find a little filament and it's going to start carrying things around. If it's part of your muscle, it's now going to be incorporated into your muscle. Whatever it is, it's now ready to do its job. If it's a hormone, it's going to go to the Golgi apparatus, get packaged into a vesicle. That vesicle is going to go to the cell membrane. It's going to be exported outside of the cell, go into your bloodstream, and be a hormone. Whatever it is now ready. We have the end process of protein synthesis. And that last phase is called termination. So termination, let's move that over. That is very rude. Please don't do that. You are not allowed to yawn loudly in class. When you do so, you communicate to me that you are not paying attention, which is bad. But also that I am boring and I prefer not to know. Sad.
That was Julian.
What?
That was
I thought-
Julian.
Oh, Julian is telling me it's Julian. That's sus.
It's pretty sus,
It
dude.
was pretty sus, no cap. That's a ligma move.
Ooh, that's a ligma move, dude.
The fruit.
That's like a
is
property.
Have you written that down? Drawn a new diagram?
Wait.
It's like a chop.
Termination.
Yeah, termination. You have to draw it with no ribosomes around. And you can draw the ribosome sort of floating in space away from everything else.
We draw a sad ribosome.
Do you still draw?
Ooh, rotated. Sad ribosome.
Doesn't the bottom of the ribosome go with it?
No,
Wait, so
no.
we don't-
Any small subunit. It's not like it owns that particular
We
small
don't- we don't
subunit. It'll
draw-
just find a different one.
Do we not draw the AUG?
Why is the NC
No, you don't need it anymore.
Why is-
They
oh,
go away with
sorry?
termination.
I don't know what you're talking about. Thank you.
But there's no way I can't add dead in front of the firefighters. That ain't easy.
It ain't easy, dude.
It ain't easy being green.
Could you make an appeal for us, though?
Say, these are the greatest gentlemen I've ever seen.
These are incredible actors. They'll be able to create a very realistic.
These are the greatest gentlemen ever seen in Privet House.
What even is this for?
No talking. They will show up. They will talk the whole time about something completely different.
Whoa.
Yep.
That's not what I should say. Prove to me.
What?
You want me to write an appeal? I need to hear some erudite questions, perhaps.
I don't think anyone
Oh.
Why is that spinning?
All written down? Good to go?
See, I'm on top of it.
Yes, Riley.
Is it okay if mine looks like a tiny bit different?
Oh yeah, because everything floats away. You can draw it wherever you want. Yeah. And it's not like one and done. That messenger RNA is going to get used again. The ribosome is going to be reading something else, maybe the same one if it starts at the beginning. The tRNAs are going to go fill up with their amino. So all the little wait staff are going to go back, the tRNAs, and fill back up with their correct amino acids and get ready to go again and again and again and again. And that is happening so quickly, thousands of times per second on multiple mRNAs in all of your cells simultaneously. And if any one process of that goes wrong, consistently, you die. Yes?
What happens if it doesn't terminate? If
It goes forever, endlessly into the void.
But like, there wouldn't be enough messenger RNA because the termination, that's an end stop.
Oh, if it just ran, there was no termination.
Yeah.
Like there was some sort of mutation where it didn't have a thing. It would just get stuck there, and probably eventually, if it was permanently stuck, eventually enzymes come around like that's not working. They chop it up, and it becomes part of the waste of the cell.
Interesting.
Yeah.
How are dead cells
Fascinating.
expelled from your body from life?
Pardon? How or what?
Dead cells expelled.
They go into your bloodstream, chopped up. Your bloodstream carries things to your kidneys, and then the waste is excreted. So, for example, if you, one of the worst eating disorders, rowing. You have to hit a certain weight class, let's say, for rowing. And so for beforehand, you're been told by your coach, this is incredibly unhealthy, not to eat. And so you're reducing your weight.
Understand.
Where does the fat and protein that you're breaking down go when you're losing weight? How do you get rid of it?
Sweat.
Pardon?
Energy, burns into energy.
Burns, some of it burns into energy, so you literally breathe it out as carbon dioxide. The parts you don't breathe out, and that's especially the nitrogen parts, get turned into urea, which gets filtered out by your kidneys in your urine.
What about sweat?
I did encounter a student once in that process. They thought they pooped their fat out if they went on that. You don't poop your fat out. You have to literally burn it. Burn it or pee it. Yes?
Doesn't sweat also work?
There is some urea in sweat. In fact, you can think of sweat as just sort of pee that you excrete out your skin.
Because
It
I
also has
understand it
urea in it, which is why your clothes will go yellow over time.
Yeah.
You can add bluing agent to your clothes to like make them look pretty again, because your clothes will slowly go yellow with your sweat. So when someone gets their sweat on you, it's like a kind of dilute pee. It's not as concentrated as pee, but it has all the same stuff in it. It's just more water.
It's like
Yeah.
if a UFC fighter needs to cut weight, he goes into the sauna with a thermal.
Oh yeah, and that just gets that type of weight loss isn't actually burning any fat. What you're doing is you're sucking all the water out of your body. So if you it's not recommended that anybody regularly weigh themselves. That leads to disordered eating. However, if you have ever weighed yourself consistently day by day, say under doctor's instructions, you will notice your weight can fluctuate by 5 to 10 pounds per day sometimes. Now, 10 pounds would be unusual. It depends on your particular size. But that can all just be water. One day you didn't drink enough water. 2 pounds, a liter of water is 1 kilogram, 2.2 pounds. And that amount in your body can fluctuate. And the taller and larger a person you are, the more that can fluctuate because you just have more room for water to expand and shrink. Is everybody done?
Yes.
Everybody still ready? Okay, turn to the next page, which is this. I would like you to use your brand new table, take the mRNA, make the tRNA. Now, this doesn't start with the correct one. It should be AU. What's the correct starting codon? Pardon? Yeah, and that's because when I go on the internet, I just stole this off the internet from another teacher. Found it on a web page. I don't really think it's stole because they're like anybody can use it. But people, when non-biochemists make these, they don't necessarily think like it should start and stop. But when I make one, it will start properly. So go until you get a stop codon, or just go until the end if there is no stop codon. We'll pretend this is in the middle somewhere, and that's why there's no AUG at the beginning. Pardon?
You just make the C a G?
No, you can't, because that's what's printed there. And if I look at it, I'm going to be looking for what I printed. Because I never make a mistake. Well, I did make a mistake once, but that's the only mistake I made. What mistake? Someone told me I was wrong, and I believed them. And it turned out I was right all along. So my mistake was believing that I was wrong.
So always
Remember,
believe you're right.
if you're confident, you can be president someday. If you are right, no, maybe, one would hope, but not always.
Stop.
You,
First one is
A,
stop. No, it
G.
isn't.
Yeah.
UAG is stop.
Dude, you look at the
What
mRNA.
would you stop?
Ah, so which one's the codon?
What?
Wait, you fell.
No, but it's renewing the mRNA.
the mRNA. So this
Come on,
is the one you look up because that,
work harder.
3 of them
Just cat splits it.
is the codon, and that
Proline.
is the anticodon. No, what does AUC code for?
Isoleucine.
No. Here, A goes to
U,
A, G, and then what goes down here?
Isopro.
You said it just a second ago, I thought.
Isoproline.
Isoleucine.
Oh, I can't read. Stupid.
And did you look up AUC or UAG?
AUC.
AUC. That is the lookup because that's the codon. And I put this example deliberately this way because I know some students are going to go, oh, and they look at this one and then code it. And when you get used to, that's not, it has to be the mRNA. There are tables, I've worked in a biochem lab, and there are anticodon tables where every amino acid is given by the anticodon because that happens to be what we're working with, and it's way easier to quickly look it up. To be honest, once you do this long enough, you have them all memorized.
Really?
It's not that. You ever go to a grocery store and you're like, oh, I have the celery, and they don't have to look up the code. They just punch it in. If you do something all the time, you are actually pretty good at memorization. You're actually probably at your best right now. You've got nice, young, healthy brains. If you ever want to memorize a huge amount of facts, now's the time to do it. 'Cause you'll slowly decrease your ability to do it. But even old people can do that. Yes, Jojo, you had your hand up. Can't find.
First 3 letters.
CAG.
First 3 letters. Oh, AUC.
Hold it. Did you have all the other ones?
You're wrong.
Show me. Show me.
What'd you say? You're wrong. What do you mean you're wrong? AUC was a snob. Did not. Look right here, buddy. Oh, I read it wrong. I read it as, no, it's UA. Oh, shoot. Oh my god. Get all of this guy, am I right? I read it wrong. Hey, that one's wrong too.
Going down.
Nope. Yep. GGU.
You take the first 3 letters. Is that like how it
It's glycine.
One
Oh,
more.
my fault.
Oh my goodness. Oh my goodness. What an upset. What an upset.
Go back. Go back.
Tottenham for the win.
Go back. Go back one more. It should be on that page, which is so weird. Sometimes I notice the Adobe printer, like that makes PDFs, doesn't deal well.
You are
sometimes
stupid.
with it. That's how you print it. Yep.
This is just
Question?
as a
Oh, we're done. So where's the
Isoleucine. The top one.
And before I do that printing, I didn't mention the whole thing down here. What's this? Has anybody been using this? This is identical to your table. I included on here because what type of table you get will depend what university you go to and what your biochem professor likes or biology professor. This is the same as your table, and some students like it a lot more because you just start with your first letter. So A,
Oh,
U, C, isoleucine. You have UAG, U, A, G. Stop codon. Stop. So this is another way of organizing the data. I really like this. I was going to use it on our data booklet, but some other teachers preferred the table one. And it's just what you're used to. And so you can use either. You'll get the data booklet on your quizzes, so you should know how to do that. But if you prefer these, like there's tons of ways to do it. If you want to come ask me a question, I'm going to print something for JoJo. I'm going to try and print just this page.
Look at my answer. Are you done this?
Just to do this.
That's what I thought.
Yeah?
is?
Oh, stop. Yeah.
Right, stop.
You just write stop.
No. Question. How do you get the abbreviation of it?
What?
Oh, I guess we have to go, Noob.
I thought class ends at like 12,
Actually, no, we can
12,
stay.
10.
I don't really want to go to.
I didn't hear a bell, remember?
That's
If I
fine,
don't
that's
hear a
fine.
bell,
That's fine
I
with
teach
me.
forever.
That's fine with me. I'm one of those socials anyways, right?
Person.
CAG.
Jojo, if I can get a PDF of that page that works, I'm going to just email it to you.
Dude, why don't you just do it later? Because I'm not gonna do it later.
Nope, you could just tell him you don't want to do it today. You won't make you do it today.
I'm done. I'm actually so like, you don't understand how shifty I am. Like, I'm pretty much raw Dylan.
You're shifty.
I am shifty.
Shifty.
I am
You should
Shifty.
tell what's his name, Chris.
I'll tell Mr. Chris. He's
Say,
shifty.
We're both very shifty people. We're good actors.
We're good actors.
We work really well in this
And we make the
simulation.
play.
We make the play.
We make the play.
Are you in acting?
I would like to call myself a jack of many
We are
trades.
actual. We are dancers. We are.
I
We
like
do
that.
dance.
Like,
We do
oh.
dance. Noof, come on, Noof. We know
Ponder.
you're good
We
for this.
ponder, Mr. Newton.
You remind me, I was once on a train with a friend, and the conductor came down, and they're like, tickets please, and like we didn't have any, we were told we could buy them on the train, and he goes, yeah, you can, and I said I'm a student, and I was, and I paid a lower price because a student, and then he goes to my friend, oh, are you a student? And the guy goes, who's my friend, who's not a student, is like, yes, the guy starts to make the ticket, and then he goes, of the occult, and the guy goes, I've already made the ticket, yeah, fine, you're a student.
Of the what?
Of the occult.
Noof, come on. You know we'd be really good for
Were
this.
you in a
met
cult?
The word? Occult? Oh,
Wouldn't be
great.
surprised.
You've learned a new search term. Black magic.
Wait, is CRISPR a cult?
I would like it to be a cult.
Noof, if he says he can get a few more people for Privet, could we be first on the list?
You can. He's sick.
We
Come on,
should make
come on,
Privet
Noof.
Squid Game.
That would be so cool, Noof.
Oh, hey, come up with a Privet Squid game.
Can we
I
run
mean,
that?
when you're in grade 11 or 12, you'll be running the house. God rest our souls. No, I don't want to touch people who are sick.
I'm
Go
not
away.
sick. I'm not
Let me
sick.
go away. No, you might be sick. You've been near
Elbow
here.
touch, elbow
Elbow
touch.
touch. Elbow touch.
Hey.
Um,
Someone forgot their phone, Noof.